So, hello, everyone. It's a great pleasure for me to introduce Dr. Sheng Zhong from the Department of Biomedical Bioengineering. His talk is going to be about single-cell chromatin and RNA interaction in the aging human brain. Thanks.
Thank you, Matteo. Thank you, everyone, for coming. So when I look at the very nice poster that is shared for you, so I have a nice photo with a nice piece of pizza beneath me. That reminds me acronym of a PDE. It's not a partial differential equation. It's a pizza delivery expert. So I wish to share with you this work titled Single-Cell Chromatin and RNA Interactions in Aging Human Brain. I'm a professor in the Sheng Department of Bioengineering. I'm also a faculty member of the Bioinformatics and Systems Biology program. So there has been a tremendous amount of novel knowledge accumulated in the recent years about how the genome is organized in 3D in human cells. So we now know that the chromosomes are typically folded in a way that if you look at any small pieces of the nucleus, you might find contacts of pairwise chromatin interactions or multiplex, multi-way interactions. These interactions in many single instances are known to regulate transcription of specific genes. For example, the looping of enhancer to promoter brings enhancer and promoter in proximity, which utilizes the enhancer sequence to regulate the target gene of the promoter. Many other proteins may facilitate this process. There may be transcription factors or chromatin-associated RNA that facilitate the looping of enhancer to promoter. Now that we also understand that there are many RNA species actually associated with the chromatin, now called chromatin-associated RNA, which regulate gene expression in various ways, including, for example, recruiting epigenetic modulators to the chromatin, including serving as scaffold to keep other proteins on the chromatin or directly participate to tether enhancer promoter looping. Even though there are many individual cases of how singular genes are regulated by the 3D structure of the genome and the associated RNA, in general, it remains debatable whether the 3D genome organization has a widespread impact on the expression of numerous genes. How to address that question? So there is a proposal titled Single-Cell Multimodal Omics, the Power of Many, published as a perspective, as a comment in Nature Methods 4 years ago. So the gist of this comment is that perhaps when you are able to simultaneously observe the structure of the genome and transcription from the same single cell, perhaps there is a way to tease out whether the genome structure has a widespread impact to the expression of many genes. So the search for a single-cell multimodal, multi-omics technologies has begun many years back, perhaps 10 years back. So there are tools that would enable seeing, observing multiple molecular species or multiple genome structure aspects from the same single cell. For example, listed in columns, including CARs, these are chromatin accessible regions, or histone modifications, or DNA methylation, or 3D genome organization, or gene expression, or RNA chromatin association. And this is only a partial list of the omics and imaging tools that are able to simultaneously capture more than one modality of data from the same single cell. So I would like to put more emphasis on the tools that are able to capture 3D genome organization and gene expression. There are a few very useful imaging-based tools, including DNA-seq-FISH Plus or CAR-MINA. There are now a couple omics-based tools, including a high-res that is able to capture pairwise chromatin contacts and gene expression from the same cell. I wish to introduce in this work the music technique that is designed to capture multi-way interactions, including pairwise interactions of the chromatin and gene expression, as well as any chromatin-associated RNA, as well as their chromatin association loci throughout the genome from the same single cell. So music stands for multi-nucleic acid interaction mapping in single cell. So the rationale we had was that a chromatin complex can be dynamically organized to harbor multi-way chromatin interactions and or chromatin-associated RNA in that complex. So the design goal of this technique is to enable concurrent profiling of gene expression, co-complexed DNA sequences, and RNA chromatin associations from the same nucleus. To break that design goal down into very specific goals, we hope to generate a sequencing library that contains the following information. It should allow us to sequence RNA and fragmented DNA together in the same sequencing library. It should tell which RNA sequences and which DNA sequences came from the same nucleus, thus resolving single-cell information. It should be able to tell from the sequencing data whether the source molecule was an RNA molecule or a piece of DNA. It should be able to differentiate that. And it should be able to identify any DNA-DNA or RNA-DNA co-complex event. If there is multi-way interactions, it should be able to tell that there are multiple DNA pieces that are co-complexed into the same chromatin complex. So to implement that design goal, Dr. Cui Nguyen actually had this initial idea when he was a PhD student, close to his graduation in my lab. He had this idea. And now that Cui is a president fellow at Stanford University, and followed by Dr. Zhifei Luo, who implemented the strategy when Zhifei was a postdoc in my lab. So the strategy works as follows. So first, we would like to attach a unique piece of sequence to fragmented DNA in the nucleus. That piece of sequence we call as the DNA linker sequence. And we wish to attach in situ a unique piece of RNA sequence, a defined piece of RNA to any RNA molecule. And later, when I sequence out the sequencing library, I could read from the DNA linker or the RNA linker to know whether the original molecular species was DNA or RNA. So we cross-link the cells to preserve any chromatin complexes, we fragment the genome, and then we ligate DNA linker and RNA linker, respectively, to DNA and RNA in situ. And next, we use this split pool strategy to add single-cell barcodes to any DNA or RNA. So this is a relatively standard technique that would enable you to split and pool and iteratively add up to several rounds of single-cell barcodes that we use 3 rounds. So those barcodes will be added to both the DNA and the RNA. And later, the sequences sharing the same cell barcodes were determined to come from the same cell. And at this moment, we would harvest the nuclei, lyse the nuclei to release molecular complexes, and we wish to add single nuclear complex barcodes now. So this is achieved by inputting those released complexes to the 10x microfluid gel bead system that use tiny gel beads to capture groups of molecular complexes, which will then add it with the same 10x barcodes. And finally, we harvest those complex barcode added material and do another round of split pool and split in this way, split into as few as only 8 tubes and add a final round of split tube barcode. So that is meant to augment the capacity of 10x gel barcodes coding capacity. So in the end, what we have is a sequencing library of the following. It has an insert in between. This insert may come from RNA or DNA. Next to the insert is a piece of linker, which we know by sequence whether this is an RNA linker or DNA linker, allowing us to differentiate whether the insert was RNA or DNA. And then it would have a set of 3 cell barcodes. Collectively, we call the cell barcode. Using that, the sequences sharing the same cell barcode would be determined as coming from the same single cell. And finally, there's a set of 2 types of complex barcodes, 10x and I7 barcodes. When the sequences share both the cell barcode and the same complex barcode, we determine that those sequences came from the same molecular complex of the same cell. And so in the end, so the sequence configuration would be as follows, as I described. So we decode this sequence configuration. We expect to find the cell barcodes, complex barcodes, RNA linker, or DNA linker. That enable us to tell all the design goals as we described in previous slide. So we put this into a test. So we used this typical design of cross-species experiment where we mix human embryonic stem cell and mouse embryonic stem cell in a mix. And we carry out music on the mixture of the cells with the following purpose. In the end, if we determine any sequences from human and mouse to come from the same single cell, it means that it's not a real single cell technique. If we determine any sequence from human and mouse to come from the same complex, it means that's not really single complex technique. So in the end, what we find is that the cross-species mixing rate for the single cell test is 0.91. That is on par or perhaps beneath the typical single cell threshold of judging that most of the reads were indeed coming from the single cell. And at a complex level, the cross-species mixing rate is even lower, 0.13%, meaning that most of the contrasts that we infer come from the single complex. We actually anticipated that the cross-species mixing rate at complex level is lower than the cross-species mixing rate at cell level. This is because for any 2 sequences to be determined as coming from the same cell, they only need to share the same cell barcode. For them to be determined to come from the same molecular complex, they need to share both the identical cell barcode and the identical complex barcode. So therefore, we anticipate that at complex level, the mixed cross-species mixing rate would be lower. So in order to test whether this technique would be able to recapitulate what we expect to see in terms of the genome structure and gene expression, Xin Wen, who is a 5th year BISB PhD student who's close to defend her thesis, did the following tests. First, she took the ensemble of music data from single cell to make it into ensemble and compared that to the bulk micro-C, a technique to capture chromatin structure from the bulk. So on the top right corner is the interaction map of one human chromosome 1 from the bulk micro-C. Lower left is the ensemble of single cell from music. This side-to-side comparison led us to visually appreciate that most of the bulk structure from micro-C seems to be recapitulated in the bulk music. In terms of gene expression, so we first compared the bulk nucleus RNA sequencing showing on the y-axis of all the measured genes and the ensemble of music's RNA reads on the x-axis on your left. So we see a strong correlation between the ensemble of RNA reads from this single cell technique to the bulk measurement of the nuclear RNA sequencing. And if we go into specific strands on the right, there we have two top lanes showing the bulk RNA-seq and the bulk IMRG. IMRG is a technique that would enable us to tell the nuclear RNA. You see there's good correlation between those two bulk techniques. Beneath that is the ensemble of the music data. You see that recapitulate even the boundaries of where the expressed transcripts are. And then we can go down to single cell. There are 3 examples of single cells where we see single cell reads. And there is antisense transcript nearby that we can see the antisense transcripts at the bulk, at the ensemble of music, as well as in single cells. So this data suggests that there is a strong correspondence of single cell information, also a good resolution to decipher the strandness of the RNA, the sense of the RNA. And so if we check a known example of where RNA is expected to spread across its transcription neighborhood to demarcate a silencing neighborhood, this KCNQ1 OT1 gene, an imprint gene, is a good example. There, on the top lanes, we show the paternal allele. And on the bottom panels, we show the maternal allele. In this particular case, we see that this transcript, this imprint transcript, is expressed from the negative strand from the paternal allele. This gene is on the negative strand, where it's not expressed on the maternal allele, which is expected. This is known. And furthermore, if we check out the DNA-DNA contacts from the 2 alleles, visually, I can't note any apparent difference between the paternal and the maternal allele if we look at DNA-DNA contacts. However, if we were to check out the RNA-DNA contacts, we see that from the paternal allele, there's plenty of RNA-DNA contacts that comes from the RNA of this particular gene and demarcate and decorate its transcription neighborhood. And that type of decoration actually is known to be helpful to suppress the other genes in that neighborhood. So this is another sanity check, which allows us to appreciate that the expected RNA chromatin association information is also captured from the music technique. So to summarize for the time being, music concurrently provides DNA-DNA, RNA-DNA interactions and the transcriptome from single nuclei. The ensemble of music single-cell data recapitulate the expected patterns of the 3D genome structure, gene expression, and RNA genome association. So with this technique validated, so Zhifei went ahead to generate a single-cell map of gene expression and RNA-DNA chromatin complex map from human brain. There, Zhifei generate data from a total of 14 human prefrontal cortex samples, including 7 male and 7 females. All those folks are aged 59 or older. So this is a elderly population. And there, we resolved approximately 9,000 single cells. And using the RNA reads alone, we can cluster these single cells. And turn out that these single cell clusters were mapped to known cell types very well, including some subcell types are deciphered by the RNA reads, suggesting that the RNA read seems to carry very informative information about the cell type and even the subcellular states of a known cell type. So we see is now that we can examine the corresponding genome structure of every single cell. Now, every single cell, we know its cell type through the RNA of this single cell by clustering. So the first thing that could be examined is this type of information where every column is a single cell. the single cell are grouped into major cell types, including, for example, EXN, excitatory neuron, and oligodendrocyte, OLI. So the columns are now genomic distances, and the color density is the contact frequency of genome interactions of the given genomic distance on the row in the single cell on the column. So if I were to take a single cell, let's say the first rightmost single cell of oligodendrocyte, for example, what we see on this column is that at relatively closer genomic distance, for example, 10 kb to 100 kb, I would have a darker color, suggesting more chromatin interactions happen in that genomic distance. As I go to longer and longer genomic distances, such as 1 megabase and longer, 10 megabase, I would have in this single cell fewer and fewer contact frequency of pairwise chromatin contacts. This is very consistent with the knowledge learned from the bulk since the initial invention of the high-C technique in 2009. The first observation, among the first observation, is this reverse correlation. That is, the closer the linear genomic distance is, on average, the higher the chromatin contact frequency is, and the further and the lower, and so on. So this is very much established and expected from the bulk. And this is indeed recaptured by a subset of single cells, which leads to a surprise here, where I'm using oligodendrocyte as an example, but in every cell type, it looks like that at least in this group of elderly people, there is indeed a subset of single cells showing higher contact frequency at lower genomic distance as expected from the bulk. But there is always another subset that violates this principle that has been known for more than 15 years now. So that seems to be the cells that have lost a compact local chromatin structure that caught our eye as a surprise. So to see what happened there, the first thing we did is we used a very simple rule to dichotomize single cells into groups, two groups. So the orange group are what we call local chromatin structure eroded, LCS eroded group. Essentially what it means is that their most frequent contact frequency appears somewhere large. As opposed to the other group are local chromatin structure preserved, or the expected group, which we call LCS preserved group. So what are the other characteristics of these two groups? It was very fortunate that around the fall-winter quarter of 2023, there were a couple of pioneer papers that started to calculate something called a single cell's transcriptome age. So what they do is that they take the single cell transcription data and use a combination of the genes that are associated with aging to compute a single index, and using that index to indicate whether this single cell is relatively older or younger than the other single cells. So essentially, by using the average older and younger tissue, whether they learn a model of what an older tissue's transcriptome should look like, then they extrapolate that model to infer single cell transcriptome age of older and younger. They have this so-called single cell transcriptome age metric pre-calculated as a model offered. So then we plugged in those single cell RNA transcripts that we got to calculate for each column, each single cell, a single cell transcriptome age. What we observe, I'm taking the oligodendrocyte as an example, the brighter ones are the older single cell transcriptome age indexes, and the lighter ones are the younger ones. So what we see here is that there is a correspondence between the cells with preserved local chromatin structure to be transcriptomically younger, and vice versa. This started to suggest that there might be a single cell chromatin structure of usefulness for those single cells. So the next thing that we were able to check is that among those individuals, some of them have some pathological indication of Alzheimer's disease, namely their Braak stage are 4 or above, whereas the others would not have that indication. So we can, of course, color every single cell from whether there is Alzheimer's pathology. So we see that there's more Alzheimer's pathology single cells in the group that seems to have lost compact local chromatin structure, indicating that there might be a correlation between the loss of the usefulness or compactness of local chromatin structure and the pathology of the individual. And in terms of sex, what we see seems to be cell type specific. In oligodendrocytes, it looks like that the males are more enriched in the younger population, whereas the females there's more enrichment in the older population. In mouse study, it's actually known that the older mouse, the females start to have more dying oligodendrocyte than the male. But this seems to be the first time we seem to have an indication of this indication in humans. It's actually reversed in excitatory neurons. You see that in excitatory neurons, the healthier local chromatin structure preserved single cells are more in the females, whereas the males seems to have more of accumulation of this chromatin structure lost single cells. So with that, I wish to have a quick summary. So chromatin structure reflects single cell age and disease status. Brain cells exhibit variable degree of loss of a compact local chromatin structure. These LCS eroded cells exhibit an older transcriptomic signature. Alzheimer's brain has more LCS eroded cells, older, than the control brain. In oligodendrocytes, female has more LCS eroded, older cells than the male. So with that, I wish to ask you a question. So this is a circles plot drawn based on some data. So my question is, what do you guess is the underlying data used to draw this circles plot? So it's the melody from Four Seasons by Antonio Vivaldi. So with that, I wish to segue to cis expression quantitative trait loci, cis-EQTL. So these cis-EQTLs are genomic sites where personal genomic sequence variations explain the expression variability of a target gene. Okay. So there's a correspondence of a cis-EQTL to a target gene. And the personal variation of one explains, to some extent, the individual variation of the other. So what's new recently is that in 2022, there's a consortium effort that have systematically mapped cis-EQTLs from human brain, resolving approximately 500,000 cis-EQTLs in human brain, as well as their respective target gene in every cell type. The surprise from that paper was that most of those cis-EQTL target gene correspondence are cell type specific. So we took their data and redrawn it on the right. So every data point is a cis-EQTL target gene pair. So the total number of cis-EQTL target gene pairs are the row sums. Those are approximately 500,000 there. And the columns are the overlaps. The first 5 columns, first 6 columns are non-overlapping. Those are cell type specific pairs. And after the first 6 columns, those are pairwise overlaps shared between 2 cell types or 3 cell types and more, right? So, so the first several columns are those cell type specific ccQTL target gene pairs. As mentioned in the original paper, that is actually the majority. Why this is a surprise? Because after all, most of the single cells of the same person share the same genome. After all, most of the genome of different cells of the same individual is identical. So most of those ccQTLs would be the same genomic sequence across different cell types of the same person. Why? They would have a cell type specificity of what target gene expression to influence only in a cell type specific manner. That seems to pose a conundrum that couldn't be appreciated for the time being. And so leveraging the music technique, we took a look at this paradox. We started with some case studies. I took this NEL1 gene, it's a cell surface receptor gene, for example, and they previously mapped ccQTLs were known to only affect the variability of this gene in excitatory neurons. So showing here, so the EQTLs only pair with this gene or affect this gene expression, so variability in excitatory neuron. Another example, this PREX2 gene, a G protein in P10 signaling and PI3K signaling were only affecting the individual variability in oligodendrocytes. So we those 2 case examples. In the first example, those are the EQTLs shown in blue, and those EQTLs formed chromatin contacts by single cell music data to the promoter of the NEL1 gene only in excitatory neurons. So remember, no chromatin contacts detected in any other cell types in this data set. PREX2, there is a group of 6 ccQTLs that previously said to only impact PREX2 expression variability in oligodendrocytes. Chromatin contacts between those ccQTL loci and the promoter of the target gene were only found in oligodendrocytes, not in the other cell types. So these individual cases makes us wonder whether this is a generalizable phenomenon. In general, whether the chromatin contacts between the cis loci, the variability of that across cell types explain why the same sequence only affect variability of the target gene in a cell type specific manner. And to test that, we leverage this association test where we categorize the chromatin interactions by the observed cell type in excitatory neuron, inhibitory neuron, and so forth, shown on the columns. And there, we do not exclude any contacts. Right? And on the columns, we categorize any EQTL target gene pair in what cell type they were detected from. So that gives us the columns. And then we can test whether there's association of any cell type, whether it's association between the chromatin contacts and EQTL target pairing. This turned out to be enriched for every cell type. So it looks like that what we saw previously were not individual cases, but generalizable. In general, there's more supporting chromatin contacts in that particular cell type corresponding to the cell type where the cis-EQTL target its target gene. To summarize, cis-EQTL often affects gene expression in specific cell types. EQTL target gene pairing correlates with DNA-DNA contacts in the corresponding cell type. This suggests a widespread impact of 3D genome structure on the expression of numerous genes. The impact is not the same. It's not seen on the same cell. It's seen only when we take the consideration of individual expression variability and cell type variability, then the impact is seen in a global manner. So lastly, so I know that many folks are curious about sex difference in gene regulation, especially this mysterious X-cyst link RNA that is known to silence one of the two X chromosomes in female in order to compensate for the two X chromosomes in female. So it remains to be a current topic of whether such silencing typically observed in undifferentiated cells are indeed the case and prevalent the case in terminally differentiated cell types, especially in human tissue. And we took advantage of this data set to examine this question. The first plot shows the X-cyst link RNA expression level in every single cell, in every single brain cell, in every cell type in the columns, in female in blue and in male in yellow. Fortunately, the males still do not express X-cyst RNA in any single cell, in any cell type. Females do as expected, but not every single cell. We see in females, a subset of single cells of every cell type seems to, at least in these elderly people, remain X-cyst expressed. The other subset music technique failed to detect any. And the next question is, does the X-cyst RNA still decorate the X chromosome and specifically decorate the X chromosome as opposed to the autosomes? On the right, we plot this RNA DNA contact map. The columns are transcription loci. The rows are where RNA are transcribed from the beginning to the end of the X chromosome. And the columns are the target genomic locations of the X chromosome. A color in a given cell is how much RNA chromatin interaction is identified from the RNA transcribed from the row to the target genomic site on the column. What we see from this contact map is that most of the interactions seem to align to the diagonal line, suggesting that most often RNA chromatin interactions happen near the transcription loci. It's transcribed, it touches in transcription neighborhood genomic sequence. You see that some genes spread a little further with one very special case, that is the X-cyst gene. This transcript of the X-cyst gene decorate the entire X chromosome, suggesting that in these elderly females, it's still the case, at least from the bulk. And next, at the bottom, we calculated X-cyst to target genomic sequence attachment level throughout the entire genome. And we see that there's much stronger association of the X-cyst transcript to the entire X chromosome. There are some association to other autosomes as well, but with a clear preference toward the X chromosome. So when we submitted the manuscript for review, we didn't know whether those attachment to autosome were biologically meaningful or simply noise of the technique. We didn't have a way to differentiate them, and we let it go to for review. And in one presentation I gave, actually, I was asked, do you think those were functional or those were real? Now that there are two very recent papers suggesting that indeed the X-cyst RNA can go to autosome and silence autosomal genes. So the question that really of interest to us was whether every single cell retains X-cyst X chromosome association. And for those that do not completely retain, or do not completely retain what happened to the transcription of genes on the X chromosome. So what's shown here, every row is a single cell. And the columns on the left are the entire genome. The red places are where the X-cyst RNA in that single cell on the row has association with that part of the genome on the column. We see that for the top small set of single cells, we have strong X cyst and X chromosome association there in red. For many of the other single cells, there's not much detectable, at least in these elderly folks, and there's some sporadic association in the autosome as well. On the right, we plot a total of approximately 700 genes on the X chromosome that were previously found to have ever escaped of X cyst, X chromosome silencing in any human tissue. So those were genes found in any human tissue ever expressed from the supposedly silenced X chromosome. The color of that row of every column represents the row is single cell, the column is a gene. Those were approximately 700 columns representing approximately 700 X chromosomal genes in the order of their sequence on the X chromosome. The color represents the expression difference of this female cell on the row to the average expression of the same gene of all the male cells of the same cell type. So the color is a normalized expression level of this gene on the column in the cell on the row in this female cell. So what we see here is that as we go down from the top to the bottom, as X chromosome association decrease for the majority of X chromosome genes, at least the majority of ever reported escapable X chromosome genes, they become higher expressed in those female cells, with the exception of the X cyst itself. The X cyst itself, of course, the more expressed, the higher the chance and correlation is its association with the X chromosome. So with that, we see that in female human cortex from those elderly folks, X cyst remain by large selectively associates with the X chromosome. Good news. The loss of X cyst chromosome X association in single cells is correlated with the activation of the chromosome X genes, and there are many of those genes that are correlated with various types of diseases, and many of those show sex bias. So that I hope that I summarized the work from Xinzhao Wen, Zhifei Luo, Wen Xinzhao, Ricardo Cangelli, Cui Nguyen, Xue Yuan, Zhong Richard, that appeared online a couple weeks ago. So I also wanted to take advantage of this opportunity to introduce what our lab does. Professor in bioengineering, I had my applied math degree in 2001 and biostatistics PhD in 2005. Now that the lab has the following two research goals. On the biological side, we wish to resolve the cell atlas at a single cell resolution of all the molecular species, their quantity, and their interactions, including chromatin interactions, protein-protein interactions, protein-RNA interactions, metabolite protein interactions, protein-DNA interactions. So we wish to tell what molecular species are there, what are their respective quantities, and all their interactions with single cell resolution from the same single cell and generate cell atlas from human tissue. Our disease goal is that we hope to discover a disease mechanism, and specifically for Alzheimer's disease, and develop future therapeutics to collectively combat brain aging. Our approach is the following. So we developed new technology. So I mentioned some single cell multiomics technique. We are also developing, for example, spatial multiomics technique and other high throughput techniques that hopefully would allow us to see some phenomenon that we can't see now. And with those new technology, we wish to generate big data from, for example, human brains, from patient blood samples. And using those big data, we hope to discover mechanism, including, for example, gene regulatory circuits. And through modulating those mechanisms, we hope to develop future therapeutics. For example, small molecule-based way to modulating the gene circuits. I would like to give an example. 5 years back, we developed this silver-seq technique that stands for small input liquid volume extracellular RNA sequencing that enables folks to do RNA sequencing from a small droplet of human blood serum. Those are cell-free RNA. And using that, so we were able to fortunately work with ADRC folks, especially Edward Koo and other folks, to generate single individual longitudinal profile of extracellular RNA profile of folks that are followed at UCSD ADRC for 15 years. Using that, we were fortunate to discover RNA biomarker in the blood that not only correlates with Alzheimer versus non-Alzheimer difference of individuals, but also the temporal increase across years time span actually predicts the clinical diagnosis of the disease before the clinical diagnosis. So now that we seem to know a little bit more about what might be the underlying mechanism of this biomarker, and we perhaps could screen small molecule compounds to see if any compound would modulate that. So with that, I acknowledged several key people along the way. I also wish to mention Xiangmin Xu from UCI, Edward Koo from UCSD, Xu Chen from UCSD, Annie Hilliker, Douglas Glasgow, Xu Chen, Irene Su. Oh, I really should mention everyone there. Also our lab, so I mentioned several of them, but also that Yuan Liu and Jun Chen and many other folks, Zhang Hong, Ming, Yipeng, and many other folks are working on those directions that I mentioned to you. With that, thank you for your attention. I hope we can have a question.
So the binding of this RNA with the autosomal genes, is that somehow affected in the local structure?
That I do not know. That must be in the paper. I have the knowledge of another case, which is when the axis gene is artificially inserted into an autosome. So those cases were tested in chromosome 21 with a hope to, in future, silence the chromosome 21 trisomy by inducing axis as a silencing mechanism for the extra chromosome 21. Those efforts were pursued for more than 15 years now. And I do know that in those cases, when the axis gene was expressed from an autosome, it does change the chromatin structure of that autosome and subsequently induce silencing. What I do not know is that in the normal case, where axis travels from X chromosome to an autosome and silence specific gene, what is the specific epigenetic events? Those must be in Catherine Plath's Cell paper that appeared recently, but I have not read specifically. Yes, so the question is what specific epigenetic changes that are induced by the axis RNA? in the sequence of X-cis RNA-mediated transcriptional silencing. Is that the question I wanted to? Go ahead.
So how are you defining
So that is a very good question. So the question is, how do you distinguish this local chromatin preserved versus the local chromatin eroded groups? Right? So the difference was observed only by genomic distances versus contact frequency. No other information is used. Only information used is the distribution of chromatin contact frequency across different genomic distances. That is the sole information used, meaning the column of that matrix is the entire information used. So the highest interaction frequencies changes from the right to the left in those maps, from the shortest distance to the longest distance. We simply define a place where we simply call one the highest contact frequency is somewhere, and beyond, it's eroded when it's, yeah. That is a very good question. So the question is about single cell clustering based on the RNA reads from music. And what are those RNA reads used? So there are two types of RNA reads in there if we really have to dive into the depths. There are RNA reads that do not have a co-complexed DNA read. Right? So those would be supposedly RNA in the nucleoplasm, right? And there are RNA reads that are co-complexed with some DNA fragment. Supposedly, those might be chromatin-associated RNA, right? And so the clustering analysis, we used any RNA. We didn't specifically choose the nucleoplasm versus chromatin-associated RNA. We thought without differentiating them, those type of reads perhaps mimic single nuclear RNA sequencing the best, and the technique for using that for clustering and cell type assignment has been becoming a standard. So we only did that. We didn't specifically dissect, we can, nucleoplasm RNA and chromatin-associated RNA to see there is a difference in cell type assignment between those two types. That's a very important question. That goes back to the original inspiration of generating this type of data. That's hopefully to use this very powerful single cell technique to translate correlation into causal relationship, or infer causal relationship from correlation. Usually this is impossible. However, single cell, so if we regard single cell as an individual cell perturbation, then we actually simultaneously observing many perturbations at once. And in that case, if there is a correlation, perhaps that is more likely to be causal. No, we didn't specifically tease out in that way, but it's terrific direction to go further from this data. So maybe you can help me with this. Could you say it again and maybe yeah. I see. So if I were to repeat and in my understanding, in order to let you know whether I understand your question completely. So the question is about in this group where we call LCS eroded, whether there are subgroups in there. Right. So whether these, for example, these more extreme losses are correlated with even older single cell age, right? That's a great question. I feel that that would expand what we have analyzed. I feel it's a continuity. What we analyze essentially is the same question. It's just that our dichotomy was crude. Whether there was more detailed correlation, that's definitely worth examining.
Under promising information, see large cell-to-cell variability of large associated domains defined as large unconserved. And do you see this same kind of level of efficacy for this RNA function or the RNA function that so
that's a very good question. So we have only examined the axis example where we apparently see a high degree of heterogeneity to the extent that some single cells we fail to detect axis X chromosome association at all. So I suspect this might be the case for other RNA as well. That's a terrific question. It should be examined. We have not. Yeah.
Thank you for sharing. Thank you. Thank you. Thank you.